Hi everybody, this is Emily Taylor, and I'm going to be presenting my identification of an unknown bacteria presentation. So first I'm going to talk about the environment that I collected my specimen from. So my bacteria sample was collected from the sink handle of the middle sink in the first row of the microbiology lab. This is when I formed my hypothesis. So my hypothesis was that I believed that my bacteria would be some strand of Staphylococcus, because Staphylococcus is normally found on the skin and the sink handles are being touched by live skin. This is going to talk about my specimen collection and just how I collected my specimen. So I collected my sample in late January around 11:30. I took a nutrient agar plate and sterile cotton swab. I swabbed the left handle of the middle sink in the first row of the micro lab, and then using sterile techniques, I streaked it on the nutrient agar plate with the bacteria. Then the plate was put in incubation for 24 hours at 37 degrees Celsius. Then we did aseptic techniques. So we didn't know that we were doing this project till later, so I don't have a picture of my actual master plate, but I was able to find something that looks very similar to what my master plate looked like. So I took one of the colonies and I inoculated it onto another sterile agar streak plate, and so that is what my master plate looks like, and that is what my streak plate ended up looking like. And if you see that small white colony, that is the colony that I used for my streak plate. Looking at the isolation of my bacteria, the isolation of my bacteria was achieved by streaking a chosen bacteria and adding it onto a new nutrient agar plate. My streaking was successful, although I didn't do a great job. I did end up having 4 separate isolated colonies, so that was all I needed for it to be successful. Looking at the morphology of my bacteria, I'm first going to talk about macro characteristics, which are the characteristics that can be seen by the eyes, and then micro characteristics, which are the characteristics that can be seen through the microscope. So the macro characteristics is, first, it was yellow-white in color. It was a very odd color that was kind of a yellow-white. It was also slimy and mucus-like, and it was spread out in a colony and circular shaped. It also smelled horrible. The micro characteristics was that I found it was a cocci shape, and it was in arrangements of 2 to 3. They were very, very small, and they were in chains. So now I'm going to be talking about the characterization of chosen colony staining. So I'm going to be going through the stains that I performed on the bacteria and what the result was. So this is my chart of stains that I did. I'm going to start with acid-fast. So it was acid-fast negative, capsule positive. For the endospore staining, non-spore forming, it was gram-positive. And then when we look at negative staining, it was small spherical cocci in clusters. And then when we look at negative staining, my bad. When we look at simple staining, it was small clusters of cocci. And then when we look at different media that I put my bacteria on, I'm going to start with talking about blood agar. So when I put my bacteria on blood agar, it did not produce any hemolysis. And then the methanol salt, there was bacteria growth, but it did not ferment salt. On the methylene blue agar and the McConkey agar, there was no growth because my bacteria is gram-positive. And then the triple sugar iron agar, it was alkaline, acid, and produces gas. And then looking at the biochemical testing, it was catalase positive, oxidase negative, sulfide negative, enediol negative, methyl red negative, and citrate negative. In conclusion, based on all my test results, I have decided that my bacteria is most similar to a strand of Staphylococcus, more specifically Staphylococcus epidermis. And I have 2 pictures of the strand of Staphylococcus epidermis. Staphylococcus epidermis is a gram-negative bacteria that is one of the 40 species of Staphylococcus. It is a normal part of the human flora, and it is found most likely in the skin, but also in the respiratory. It lives on human skin, and it likes to be in wet, sweaty places, so it usually lives on your hands, sometimes like under your neck, in your armpits. It's very harmless, but it sometimes can cause infections in people who are immunocompromised and in the hospital. And its infections are usually associated with like hospital devices. So it's very common to see Staphylococcus epidermis infections in like causing UTIs in catheterized patients, or causing surgical wound infections or prosthetic heart valve infections. These are kind of common to see when we're looking at Staphylococcus epidermis causing infections. I'm going to end with a summary of my lab experience this year. Although our semester was cut short, I enjoyed lab greatly. I did a lot of things I've never done before, and I thought they were all really cool. I learned about testing methods. I got to learn about microbes that I had never heard of, and just doing the testing and being so hands-on was really fun for me. I enjoyed the process of finding my bacteria. I was super excited when I finally figured out what it was, and I'm very glad I took this class. It was just a fun class in general, and it had so many different things involved in it, and I just really enjoyed it all together. Thank you so much for listening to my presentation.